Good morning and welcome to PsyQuil. I'm your host, Cy Wraith, and this is another edition of PsyQuil Daily News. Today I would like to talk about antibacterial resistance. There is a new synthetic antibiotic that has been created to work against the resistances that some bacteria have built up against our regular antibiotics. So this article is from phys.org, and it's by Rob Mitchum at the University of Illinois at Chicago on February 17, 2024. New research helps create new antibiotic that evades bacterial resistance. This is showing the methyl groups on the functional area of the ribosome for the bacteria. This is a methyl group here, and there's two of them. So this part adds one methyl group, and then this part of the reaction adds this methyl group. And with these methyl groups, they block our antibiotic from attaching to the ribosome in this area, in this A2SO3 area, or 503 area, which would then stop the ribosome from creating proteins, and then the bacteria would die. We'll get into that below. Scientists at the University of Illinois Chicago and Harvard University have developed an antibiotic that could give medicine a new weapon to fight drug-resistant bacteria and the diseases they cause. The antibiotic, chrysomycin, described in Science, effectively suppresses pathogenic bacteria that have become resistant to many commonly prescribed antimicrobial drugs. The promising novel antibiotic is the latest finding for a long-time research partnership between the group of Yuri Polakhanov, Associate Professor of Biological Sciences at UIC, and colleagues at Harvard. The UIC scientists provided critical insights into cellular mechanisms and structure that helped the researchers at Harvard design and synthesize new drugs. In developing the new antibiotic, the group focused on how many antibiotics interact with a common cellular target, the ribosome, and how drug-resistant bacteria modify their ribosomes to defend themselves. So more than half of all antibiotics inhibit growth of pathogenic bacteria by interfering with their protein biosynthesis, a complex process catalyzed by the ribosome, which is akin to a 3D printer that makes all the proteins in a cell. Antibiotics bind to bacterial ribosomes and disrupt this protein manufacturing process, causing bacterial invaders to die. But many bacterial species evolved simple defenses against the attack. In one defense, they interfere with antibiotic activity by adding a single methyl group of one carbon and 3 hydrogen atoms to the ribosomes. Scientists speculated that this defense was simply bacteria physically blocking the site where drugs bind to the ribosome. But the researchers found a more complicated story, as they described in a paper published last month in Nature Chemical Biology. By using a method called X-ray crystallography to visualize drug-resistant ribosomes with nearly atomic precision, they discovered two defensive tactics. The methyl group they found physically blocks the binding site, but it also changes the shape of the ribosome's inner guts, further disrupting antibiotic activity. Polakhanov's laboratory then used X-ray crystallography to investigate how certain drugs, including one published in Nature by the UIC-Harvard collaboration in 2021, circumvent this common form of bacterial resistance. Quote, by determining the actual structure of antibiotics interacting with two types of drug-resistant ribosomes, we saw what could not have been predicted by the available structural data or by computer modeling. It's always better to see it once than hear about it 1,000 times, and our structures were important for designing this promising new antibiotic and understanding how it manages to escape the most common types of resistance. End quote. Chrysomycin, the new antibiotic, is synthetic. It's pre-organized to avoid the methyl group interference and attach strongly to ribosomes, disrupting their function. This process involves locking the drug into a shape that is pre-optimized to bind to the ribosome, which helps it get around bacterial defenses. Quote, it simply binds to the ribosomes and acts as if it doesn't care whether there was this methylation or not, Polakhanov said. It overcomes several of the most common types of drug resistance easily. End quote. In animal experiments conducted at Harvard, the drug protected against infections with multi-drug resistant strains of common diseases drivers, including Staphylococcus aureus, Escherichia coli, and Pseudomonas aeruginosa. Based on these promising results, the next step is to assess the effectiveness and safety of chrysomycin in humans. But even at this early stage, the process demonstrates the critical role that structural biology plays in designing the next generation of antibiotics and other life-saving medicines, according to Polakhanov. Quote, without the structures, we would be blind in terms of how these drugs bind and act upon modified drug-resistant ribosomes. The structures that we determined provided fundamental insight into the molecular mechanisms that allow these drugs to evade the resistance. End quote. So I want to go over the technical aspects of this now. I jumped in there and just read the whole article for you. Well, let's go over, first of all, what a ribosome is, okay? So we're going to go here to, yes, this one. This little browny green looking thing in the background behind this blue strip. This thing here is called the ribosome. Many of these float around in the cytosol of cells, including bacterial, and they attach to mRNA. They attach on the 5' side. All strips have a 5 and a 3' side. So the ribosome attaches on the 5' side, and then it reads these letters. These letters are actually the alphabet of life. It's a 4-letter alphabet consisting of an A, a C, a G, and a U. A U for RNA, but for DNA it's a T, thymine. The uracil is actually RNA existed before DNA, and it was more modifiable, more mutable, we say, than DNA was. So eventually RNA evolved to have a T instead of a U here, which made it much better at keeping its form and not mutating, which is essential if you have the blueprint of life. So anyway, the secret of life here, the alphabet, is A, C, G, U, or T. And it's adenosine, C is cytosine, G is guanine, and U is uracil. The T is thymine. And A attaches to U or T, and C always attaches to G. Now here, we have something called a tRNA. This is also another molecule that floats around in the cell. Once the ribosome is connected and starts reading these letters in groups of 3, it's called a codon when it's a group of 3, it'll stop and wait for a tRNA. The tRNA will then be able to bind, but only the tRNA that has the exact opposing sequence. U always attaches to A, C always attaches to G, and G always attaches to C. Okay, so it will bind if it's the right combination. You see that it's all U's here. So only the exact tRNA can attach to this particular codon sequence. And on the other side of the tRNA is a binding point for a very specific amino acid, in this case lysine. So essentially what we're saying is this nucleotide sequence of 3 A's codes for lysine. With the help of the tRNA, we actually get this particular amino acid attached to the ribosome at this position. And then the ribosome will move once it's attached. It will move on to the next one. Another tRNA will bind, and it will connect and add the appropriate amino acid for this codon sequence. And it will keep doing that along the whole thing. And what you end up with is what's called a peptide chain, a bunch of amino acids chained along in a string, kind of like a necklace. And they are essentially the protein, but it's unfolded. Once this long chain is done, it then needs to fold into the appropriate structure. Form is function in biology. It's a very important concept. In fact, it's so important in the case of these proteins, if it doesn't have the right form, it completely fails and is useless to the cell. We have something called chaperones, many types of chaperones for proteins that need to fold a very particular way. Some of them are very sensitive to the environment. So the chaperone creates a very isolated, exact environment that protein requires in order to fold in exactly the way that it needs to. It's very complicated, but I just wanted to get that out there a little bit. So this is what's called protein synthesis. This is the RNA side. This strip of RNA before it hits the cytosol will copy from the DNA, which is in the nucleus of the cell, and then flow it into the cytosol where it will be synthesized. It's a piece, but not the whole strip of the DNA, just a very particular piece. There's tons of these. Now, that should give you a good understanding of how the ribosome works. And so what happens is, with these antibiotics, they stop the ribosome from binding to the RNA entirely. But the key thing to note here is that the antibiotic stops the ribosome from binding. Okay. Then the bacteria evolved, this resistant bacteria, to have this methyl group so that the antibiotic can't bind to the ribosome. And so by blocking the antibiotic from binding to the ribosome can then bind to the mRNA and continue to do the protein synthesis. Now, what this anti-resistant antibiotic does is ignore the block that the resistant bacteria has evolved, that methyl group block, so that the antibiotic can then bind to the ribosome again, which stops the ribosome from binding to the mRNA, and then again the bacteria cannot complete photosynthesis. So it's a block of the block of the block, essentially. And this is this happens often in nature. what we like to call the arms race of nature, where there's two competing things that are often evolving against each other over time. It's very interesting. You can see it with predator and prey. You can see it in microbial things like this. It's all over the place. It's really very interesting stuff. Anyhow, I hope that explains that part of it very well. There's one last thing I want to touch on here. Well, two more things, I suppose. This woman, Rosalind Franklin, born in 1920, she was essential to the discovery of the double helix of DNA. It was her skills with crystallography alongside that ended up getting her to work alongside James Watson and Francis Crick, who got their Nobel Prize for discovering the double helix of DNA. This is the famous picture that shows the double helix of the DNA here. And here's an example of the double helix coiling around each other. And this would be where the A and the T bind, and the C and the G bind, and so on. Anyhow, so her work on crystallography and her skills is what allowed us to discover the shape of DNA. And it's a very difficult skill to have, but it's something that is mentioned in the article, and I just wanted to point that out. And one last thing, this is the book by James Watson that talks about his discovery of his double helix. You can see it here. And it's an exciting journey. It's got the history of genetics before the discovery of the double helix, the work with Drosophila and other Mendelian type genetics studies. It's very interesting, and I highly recommend it. It's one of the books that led me to performing or getting a biology degree. So I recommend it, and I hope that you consider reading that book. It will take you on quite the journey. I promise you that. Anyhow, tell me what you think about this study, this new antibiotic. What's it called? Cresomycin coming out. And please like and subscribe, and I'll see you in the next video.